This is a production of Cornell University.
So I'll be discussing my research in detecting genetic and local environmental effects using high-throughput aerial imagery. There's a long history of satellite and airplane imagery for applications in agriculture. For example, in 1972, when the United States launched Landsat-1, the satellite into orbit, that enabled a lot of remote sensing applications to begin, and for instance, the vegetation indices to be developed. However, applications for genetic improvement in plant breeding have been hindered by the cost, the resolution of the imagery, and repeatability due to weather or other positioning problems when you want to image the same field throughout the growing season. So today we have a low cost and stable platform in the form of unoccupied aerial vehicles, or UAVs. And this enables high resolution images. It's scalable in that a single pilot or operator can fly many fields in one day and could potentially fly every day of the growing season. And for instance, I flew 18 flights this summer of a field in Aurora and 28 flights of an alfalfa here in Ithaca and many others. And I use, or we use the Microsense multispectral camera, which captures in the blue, green, red edge and near infrared spectra. So you can see the camera here with the 5 distinct lenses. The thing to remember is you have to take a picture of this calibration panel prior to flying. And post-processing, I follow 2 steps. The first is to generate an orthophotomosaic using Fix4D. I want to explore using OpenDroneMap because it's an open source software. Then the second step is to upload those orthophotomosaics to imagery. So this website I have, imagery.org. So this is a Docker deployable software, so you can set up an instance on your own computer if you wanted. It's open source and it houses all the field genotypic data and analyses. So in 2019, we have this genomestofields field. This was 232 hybrid planted over 500 plots. And with Nick Casmore and Nick San Antonio, we got 6 flights. If you look closely, you can see the red and blue lines for the plot delineations. Zooming in, you can see these plot polygons, and you can see it's fairly high resolution. And each of these plot polygons is saved in the database. I went ahead and calculated vegetation indices for each plot, for each time point. So I have NDVI, NDRE, TGI, and VARI. These vegetation indices are nice because they're derived simply from means of the images. But in the future, I want to find phenotypes that can better capture features that are latent in the image. So that's for a future project. But once I have these phenotypes, I use linear mixed models to separate genetic from environmental effects. So for the fixed effect, I account for the replicate in the field. For the genetic effect, I use genomic relationships that are calculated from close to 1 million GBS markers that were genotyped for the parents of these hybrids. And the random environmental effects can either be for spatial effects or permanent environment effects. So going in a little bit deeper into the models that I'm looking at, I'm looking at 3 different classes of linear mixed models. The first class is these spatial effects. So it can be a 2-dimensional spline or an autoregressive process. And this class of linear mixed model takes information from the rows and the columns of the plots in the field. And so then the second class is these temporal effects or permanent environment effects. And they're computed using a random regression. And the third class is a random regression, but I can incorporate either spatial effects or soil survey information or any plot-to-plot correlation structure could be incorporated in that 2-step random or into that random regression. The outputs of these models are what you see on the right. So I'll have genetic effects for each line across time. So each line represents a hybrid, and x-axis is time, and y-axis is the genetic effect. And also, as you see on the bottom right, I'll have the environmental effect across time. So these are heat-like snapshots of the field, of the plots in the field over time. And the random regression has many benefits, including that it fits a continuous covariance function across all the time points. So you can essentially evaluate the genetic or environmental effect at any given time point. And it can fit large numbers of time points, which multi-trait models could have convergence issues with. So with these models, there's still a question of which one is capturing the effects truly. And since we don't know either the genetic or the true genetic or environmental effect, I can use simulation. So this is a 3-step approach where I first take one of those models and I minimize the local environment effect for using real data. Then the second step is to add in a simulated known effect. And the third is to observe which of these models can most accurately detect the simulated effect. So this is a work in progress, and I should soon have results for. And lastly, I want to look if soil survey information can explain some of the variation, this local environmental variation. And in 2019, we have electrical conductance measurements and altitude measurements that were measured by Nick Kazmar. And so on the left here, you see electrical conductance, and on the right, altitude. And we can also compute like first and second derivatives of those measurements to try to account for curvature of the soil or curvature of the field, which is known to be a source of variation. So in closing, UAVs allow for low-cost, high-resolution phenotypes. Imagery can be used to quickly extract plot-level phenotypes. Linear mixed models can separate genetic from spatial and temporal or permanent environment effects in the field. I can use simulation to probe which model best captures the true effect. And soil survey info may provide insight into the source of the effect. So I want to thank my committee, Kelly Robbins, Lucas Mueller, Harold Finesse, and Joe Guinness. And here on just these are the flights I took in this year of the alfalfa. So looking forward to analyzing that also. So thank you.
Awesome. Thank you, Nick, for that great talk. If anybody has any questions, the chat is open.
Hey, Nick, this is Jean-Luc. This is probably an impossible question to answer, but can you explain briefly the difference between the spline model and an autoregressive model? I'm kind of familiar with autoregressive, not too familiar with splines.
Yeah, so the autoregressive assumes a fixed correlation between all of the plots in the field. So if you move away 1 unit of distance from a plot, you'll it follows a correlation of, it has a fixed correlation. And if you move away 2 units, it's that correlation squared, 3 units, that correlation cubed. But with the spline, it fits a function. It solves for coefficients for linear splines. So like it's a linear equation of the rows and columns and their interactions. So I don't know if that answers, but.
Thanks. Yeah, that's helpful.
Okay, well, thank you. And yeah, feel free to email me or ask me questions.
Awesome. Thank you again, Nick. And I think
This has been a production of Cornell University, on the web at cornell.edu.